Bio 181, Chapter 14, and we're going to look at DNA and how to make more DNA. So this video, it's on a couple of experiments. The first one is by Griffith, and this was really to identify this unknown substance that is going to transform bacteria from a strain that is not pathogenic, so not disease causing, into one that is disease causing. So in this experiment, Griffith injected mice with a virulent form of a virus, a virulent form of bacteria. So it was known to kill the organism, so it injected it into the mouse and it died. Then they took a form of the virus, a different strain that was not pathogenic, that did not cause disease. They isolated that, put it into the mouse, and the mouse was happy. Then what they did was they took the virulent form of the virus, okay, and they killed the virulent form by exposing it to fire, heating it up. And then they put that bacteria in the mouse and the mouse lived. So they figured out that, okay, the bacteria is no longer capable of killing the mouse. Then this is the key part of the experiment. They took the killed form of the bacteria. So this is the portion that was exposed to the heat. So it shouldn't have caused the mouse to die, except that when they put it in along with a strain of bacteria that was previously non-pathogenic, they found that combined, they actually resulted in a bacteria that would kill the mouse. So somehow there was something in this killed bacteria that was transferred into the non-pathogenic bacteria. And so they're trying to figure out what exactly is this. So they know it could be passed between organisms, but they didn't know what it was. So the next experiment demonstrated that it was, in fact, a substance called DNA that was changing the pathogenicity of that bacteria from non-pathogenic, not disease-causing, to pathogenic. So DNA, again, this we learned about in previous chapters. It stands for deoxyribonucleic acid, right? So this is, we learned about macromolecules. This is a class of macromolecules that is made up of nucleotides. It includes DNA and RNA. So it was known that this molecule existed, that it was acidic, but it wasn't sure, is this the thing that's actually heritable and changes the proteins that are made by an organism? And so the experiment from Hershey and Chase identified DNA as the substance. It was basically between protein. They didn't know if it was protein that was the heritable composition that was going to be used for making an organism, basically the blueprint, or if it was DNA. So they weren't sure. They came up with this neat experiment, and they took radioactively labeled sulfur. So sulfur represented by S, and they use this because sulfur is a very common element in proteins. So it's found in proteins, but it's not found readily in DNA. In DNA over here, maybe I should use green just to stay with what they're showing here. So over here in DNA, okay, there's phosphorus. So phosphorus is found in every nucleotide. And so since that is found in DNA, they radioactively labeled that. And then they thought, okay, we're going to trace it through a process where we have a virus that is either labeled, the protein of the virus is labeled with sulfur, or the DNA is labeled with radioactive phosphorus. We're going to track it. They infect E. coli. So the virus, this little moon lander like diagram here, this is the virus. So the virus infects a little poor bacteria, E. coli. So here's our E. coli, and is allowed to replicate and is passed down from organism to the next. And they find that when they isolate the bacteria later, that in fact there's no sulfur, so there's no sulfur present, okay, but there is phosphorus. So phosphorus is present, so that radioactive label shows up, and they realize that, okay, this is associated with this pathogenic virus. It's found when it's infected, and so the DNA is in fact the genetic material, whereas the protein was not transmitted down, and so it's not the material. So two experiments that I'd like you to know, the Griffith experiment and the Hershey and Chase experiment.